Hi everyone, welcome back to my channel. This is lecture 2 of chapter Seeds, Structure and Germination. And in this lecture we are going to do bean seed, then maize grain. We'll study about germination and also study about the conditions necessary for germination. So let's begin with the bean seed. Mostly the bean seeds are kidney shaped. So let's write they are mostly kidney shaped with convex and concave sides. Suppose this is the bean seed. So the inner part, this part is the concave side and the outer bulky part, this outer bulky part is the convex side. So inner part is concave and outer part is convex. Okay, now let's draw the structure of bean seed. You know rajma, this is how the rajma looks. Rajma is also a bean seed. Then like this. And here is a small opening. Okay, this is the covering of the seed. Now let's label it. Okay, so this boundary is known as testa. Okay, we know that seed coat consists of two layers. Outer is the testa. This is outer. And inner is the tegmen. Okay, you can remember tegmen as in your new shirt, the tag is usually placed inside your shirt. So tegmen is the inner layer. Now, so this is the testa. This testa is protective in nature. It protects the inner delicate parts. Now, this small opening is known as micropyle. And the function of micropyle is to absorb water for the growing embryo. It absorbs water for embryo. Okay, now the second function is it is also responsible for diffusion of respiratory gases, which is oxygen and carbon dioxide. So diffusion of respiratory gases. Now, suppose our seed is placed like this. And we cut it vertically like this. So it will look somewhat like this. Okay, I'm drawing. Here we have our embryo present. So here the embryo will be in center. Now let's do all the labeling. So the outer boundary is known as testa and the inner boundary is known as tegmen. Both are protective in nature. Okay, note down this point wherever you are writing, that testa and tegmen are protective. Testa protects the inner delicate part. Okay, now these two space, this is known as cotyledons. These cotyledons are generally responsible for the storage of food. So these cotyledons, they store food as well as they protect this embryo. Store food and protect embryo. Okay. Now the upper portion of this embryo, this whole is embryo. Okay, embryo. So the upper portion of this embryo is called epicotyle. Epi means above, cotyle means for cotyledons, so above cotyledons. So the part which is found above of this embryo is known as epicotyle. And the part which is found lower in this embryo is called hypocotyle. Now, this epicotyle forms plumule which will form the future shoot. So, it forms plumule. Okay, which forms shoot. Okay, stem. Now, this hypocotyle forms radicle which will form later on root. So, this hypocotyle forms radicle which will form root. Okay. Now, here we forget one labeling, which is this opening. This opening is called as hilum. Okay. Now, this hilum, it is a spot which denotes that how this seed was attached to the ovary wall. Suppose this was the female reproductive part. Here our ovule was present. So this ovule was attached through placenta with this ovary wall. So the point where this placenta is attached to this ovary wall marks a spot on the seed which is known as the hilum. So write down for hilum that it is a spot which denotes that the seed was connected to the ovary wall through placenta. Okay, write down this point. So this was all about the bean seed. Okay, now let's study about the maize grain. Maize grain is a single seeded fruit. So it is a single seeded fruit. Only one seed is present. Okay, within the fruit. Now, the second point is this maize grain is formed due to the fusion of fruit wall and the seed coat. So formed due to fusion of fruit wall and seed coat. Okay, this we studied in the definition of grain. Okay, so it is grain, maize grain. Now, let's draw the diagram of maize grain. Let's understand all the parts. Okay, you know bhutta. So this is just a small piece, one piece of that bhutta. So this is how our bhutta looks. Okay, this yellow part is this part, and this lighter white part, you will notice in that bhutta that this is the lighter white part, which is this lower part, and in this part only our embryo is present, and this is the endosperm. So this larger bulky portion is known as endosperm. Okay, this provide food to this growing embryo which is present here, and this endosperm is also rich in starch. That's why it provides food, so it is rich in starch. Okay, now the outer boundary, okay, the outer boundary of this endosperm is rich in a protein, okay, and that layer is known as aleuron layer. So I'm drawing the outer layer of this endosperm, which is known as aleuron layer. Okay, it is rich in protein. Now, this is epithelium. Okay, this epithelium, it separates the endosperm part and the embryonic part. So this layer is epithelium. Single layer. It is single layer, and it separates the endosperm and the embryonic part. Okay, now let's draw the embryonic part. So here our embryonic part will be present. Okay, this whole portion is the embryonic part. Okay, the above this that I have drawn is known as coleoptile. Okay, this coleoptile protects the plumule, which was responsible for formation of shoot. So this plumule forms shoot, and this plumule is protected by this coleoptile. So this coleoptile, it protects plumule. Now this lower portion is called coleorhiza. Okay, and this coleorhiza, it protects the radicle which forms root. So it protects this radicle. This radicle forms the root. So this coleorhiza is protecting this radicle. So it protects radicle. Okay, and this whole coleoptile, plumule, radicle and coleorhiza are part of our embryo. This whole thing is embryo. Okay, so this maize grain is an example of monocot, means there will be only one cotyledon present. So the single cotyledon, this is whole the cotyledon. So the single cotyledon which is present, okay, which is present is known as scutellum. Only one cotyledon is present, and that cotyledon is specifically known as scutellum. Okay, it is single cotyledon present in maize grain. Cotyledon. Okay, so just remember all the labelings and the functions of each. Okay, this was our endosperm, which was providing food to this growing embryo. Okay, So that was all about maize grain. Now let's study about germination. So germination is the process of formation of seedling developed from embryo. Let's understand this definition. Here the important is seedling and embryo. Seedling means young plant. Okay. So germination is the process in which a young plant is formed, which is which developed from the embryo. So let's draw a diagram and understand this. This is the soil. Okay. This is our seed present. This is our seed present. Okay. And this is the embryo. Okay. So over time what happens? This embryo, when all the conditions are favorable, this embryo, it breaks the seed coat and it comes out. Okay. Let's draw the initial stage first. The embryo is intact. Okay. Now over time what happens? When the conditions are favorable, the embryo, it ruptures the seed coat and it comes out. Okay. Then this embryo, it forms the root. Okay. And this seed, it also comes out of the soil. See, this is how it grows. It will form root. Okay. And later on, this embryo forms the shoot. The plumule part forms the shoot. So this is how we will draw. Okay. So this young part is called as the seedling, in which the cotyledons are turning into two leaf-like structures. Okay. The plumule is forming the shoot and the radicle is forming the root. And after this, the young plant is the mature plant is developed. So this whole process is known as germination. Now let's study all the conditions that are required for germination. Means when the embryo will germinate, when it will come out of the soil and form the new plant. So let's study that. The three conditions that are required for germination are water, suitable temperature, and oxygen or air. Okay. If the seed will not have these proper conditions, it won't be able to germinate. So let's study this water. Because of this water, the seed swells and the seed coat ruptures, due to which the radicals comes out and forms the root. So because of this water, radical comes out and forms the root. Okay. This water is also important for the chemical reactions and for the enzymes to act upon the food. So it is also responsible for chemical reaction. And for the enzymes to act upon the food. The enzymes will act on the food and they will break the food to make it available for the growing embryo, and it is responsible for the chemical reactions which will take in the embryo. Now, suitable temperature. So, for germination, we require an optimum temperature, optimum, just a minute, optimum temperature of 25 degrees Celsius to 37 degrees Celsius. Okay, if the temperature is too high, means 45, 50, or the temperature is too low, like 0 degrees Celsius, minus 10 degrees Celsius, 4 degrees Celsius, the seed will not be able to germinate. Of course, there are few exceptions, but in most cases, the seed will require optimum temperature of 25 degrees Celsius to 37 degrees Celsius for germination. Okay, now oxygen. This oxygen is important for the cell division, okay, which is happening in the embryo. Okay, so this oxygen is required for cell division and cell growth. Okay, if proper oxygen will be available, cell will divide properly, it will form new cells, and our young plant will be formed, then our mature plant will be formed. So this was all about germination. That's all for today's lecture. One thing I really want to discuss is that about the importance of studies. Studies are really important in our lives. I encourage you to please invest time in studies. Of course, there is slow growth, but believe me, this study will give you everything, whatever you want in your life. It will give you respect, it will give you freedom, it will give you to do whatever you want. You can buy things of your choice, you can travel according to your choices. So please invest yourself in studies. Okay, so that's all. Thank you so much. See you in the next lecture.